Hello everyone. Welcome to the new video in preclinical pharmacology. Today we will going to discuss about catalase and endogenous antioxidant. Oxidative stress is caused due to imbalance between reactive oxygen species in the cell. In that condition, catalase play crucial role as the antioxidant. Introduction. Catalase is the enzyme that break down hydrogen peroxide into water and oxygen. Hydrogen peroxide is highly reactive hydroxyl radical which attacks on a membrane lipid and causes oxidative stress in the cell. In that condition, catalase is a crucial antioxidant enzyme that mitigates oxidative stress to a considerable extent by destroying cellular hydrogen peroxide to produce water. Normal level of catalase in the blood of healthy individuals is about 111.3 plus minus 16.5 microunits per liter. Blood catalase level decreases with the age. Female having lower catalase level than the males. Catalase activity is highest at the pH 7 and lowest at the pH 5. Location. Catalase level are highest in the liver and RBC. Discovery. Catalase is discovered by the scientist Louis Genescu Thénard. He is the French chemist. In the year of 1880, he observed that a substance in the liver tissue could decompose hydrogen peroxide into water and oxygen. Initial name of catalase is hydrogen peroxide decomposing enzyme. Latter name is catalase from Greek catalysis meaning decomposition. Structure of catalase. Catalase structure is like the dumbbell shape and terminal arm. It is form arm in the catalase structure which can actively participate as the intersubunit and form knot-like structure. This is arm is start from beta barrel half and this bend like the elbow. Then alpha helical domain and beta barrel domain. It is the central feature of catalase. It contains 8 standard antiparallel beta barrel with 6 alpha helicals. It is form cavity inside the heme group. First part of that cavity place beta barrel on the second half of that cavity corresponding NADPH binding pocket is bind as the small subunit. Then the wrapping loop. It is extended region. This is flexible region play crucial role in enzymatic catalytic mechanism. It participates in the binding and oxidation of substrate such as hydrogen peroxide. Flexibility of wrapping loop is essential for its function. Then heme group. It is crucial component of catalase. Enzyme catalase decomposition of hydrogen peroxide into water and oxygen. Breakdown of heme group. Heme group act as a cofactor, provide chemical environment for catalase to perform its function of decomposing hydrogen peroxide. Then domain connection. This is part of catalase that can be connected all the remaining part in the structure. Types of catalase. First, monofunctional catalase. It belongs to original class of catalase found in plants and microorganisms. Structure may vary in number and identified by its domain in the different species. It may essential for bacterium oxidative and osmotic stress responses. Second type, Catalase peroxidase. It is less prevalent class belonging to the second group of catalase. This is found in aerobic bacteria. Molecular weight is about 120 to 340 kilodalton. Catalase peroxidase resemble in plants but not found in animals. It protects the organism from hydrogen peroxide toxicity. Then third, pseudo-catalase. It is called as non-heme catalase, which is minor bacterial protein family with managed active site rather than the heme molecule. Molecular mass is about 170 to 210 kilodalton. It is used in treatment of vitiligo. Mechanism of production of catalase. Catalase is produced in the body primarily in rough endoplasmic reticulum, where it is synthesized from amino acid based on the genetic code from DNA. This is process involves transcription of DNA to RNA followed by translation into catalase protein. Catalase protein can decompose hydrogen peroxide into water and oxygen and protect the cell from oxidative stress. Role of catalase: break down hydrogen peroxide, mitigation of oxidative stress, regulation of cellular signaling, protection against inflammation, support in disease prevention, and modulation of immunity. Diagnostic application: bacterial infection. Catalase test differentiates between catalase positive and catalase negative bacteria, aiding in the identification of species such as Staphylococcus positive and Streptococcus negative. Oxidative stress assessment. Catalase activity level in the serum can indicate oxidative stress, which elevated level observed in conditions like acute pancreatitis and various degenerative diseases. Therapeutic potential. Catalase is explored as treatment for oxidative stress related disease including neurodegenerative disorder and diabetes by mitigate oxidative stress. Food industry application. Catalase used to remove hydrogen peroxide from milk during the cheese production and in food preservation. Therapeutic application. Cancer therapy. Strategies that inhibit or inactivate catalase could reactive oxygen species mediated apoptosis pathway specifically in the tumor cell, enhancing treatment efficacy. Gene therapy. Strategies are being developed to use gene therapy for catalase overexpression, aiming to improve antioxidant defense in tissue affected by oxidative stress. Nanoparticles delivery. Catalase can be delivered via nanoparticles targeting neurons to mitigate oxidative damage in conditions like stroke and neurodegenerative disorder. Wound healing. Catalase help to mitigate oxidative stress by breaking down hydrogen peroxide, which can impair healing if accumulated excessively. Its presence to promote better tissue regeneration. Catalase identification test. These tests performed in to identify bacterial infection in organism. Procedure. Pour 1 to 2 ml of hydrogen peroxide solution into a test tube using a sterile wooden stick or a glass rod. Take a several colonies of 18 to 24 hours, test organism and immerse in the hydrogen peroxide solution. Observe the immediate bubbling. Slide method is used as a loop or sterile wooden stick to transfer small amount of colonies growth in the surface of clean and dry glass slide. Place a drop of 3% hydrogen peroxide in the glass slide and observe the evolution of oxygen bubble. In the catalase positive, oxygen bubble were seen. It means that Streptococcus positive bacteria present in that organism culture. And in the catalase negative, no oxygen bubble seen means Staphylococcus negative bacteria are present in the sample. Role of catalase in the disease state. Diabetes. Low level of catalase can increase hydrogen peroxide concentration, which can damage pancreatic beta cell and contribute to the development of type 2 diabetes. Vitiligo. Catalase level can reduce in the epidermis, which can lead to increased hydrogen peroxide concentration. Alzheimer and Parkinson diseases. A deficiency or the malfunction of catalase is linked to the pathogenesis of many age-associated degenerative diseases, including Parkinson disease and Alzheimer disease. Current research and future direction. Catalase research focus on treating oxidative stress related disease through a innovative approach including gene therapy, nanoparticle delivery, and protein engineering. Researcher may focus on the magnetic insights, protein therapeutics, and innovation in the delivery system. Future direction. Catalase-based immunotherapy, catalase-mediated mitigation of radiation damage, investigation of catalase role in microbe health, and elucidating catalase mechanism in various diseases. Thank you.